Good evening, everyone. Thank you for attending our Spring 2021 Virtual Distinguished Lecture Series event hosted by the Dean Scholars Student Association. My name is Emily May, and I'm a sophomore neuroscience major Dean Scholar. I am so thankful to be the co-chair of this event and for all the Dean Scholars who have contributed over a past year of organizing due to COVID-19 that have made this event possible. The Dean Scholars Student Organization is a student-run organization dedicated to enriching our community through intellectual opportunities, including our annual Distinguished Lecture Series. Previous speakers include U.S. National Medal of Science recipient Dr. Susan Solomon, 2017 Nobel laureate in physics Dr. Rainer Weiss, chair of zoology at the Smithsonian National Museum Dr. Carol Baldwin, and Dr. Stephen Wolfram from Wolfram Alpha. The Dean Scholars Student Association is affiliated with the Dean Scholars Honors Program within the College of Natural Science at the University of Texas at Austin. The Dean Scholars Honors Program is founded on and emphasizes student research. Established in 1983, Dean Scholars have been awarded Rhodes, Marshall, Goldwater, and National Science Foundation graduate research fellowships to further their exploration. Our community deeply values inspiring the next generation of scientists and offering opportunities to hear from leaders in STEM fields. In addition to hosting the Distinguished Lecture Series, Dean Scholars organizes science demos at local K-12 schools, in addition to tutoring and participating in various service and leadership activities at UT Austin. Please visit our website or follow us on social media to keep informed on our upcoming events. Please note that tonight's event is being hosted by the Dean Scholars Student Association, a sponsored student organization. This is not an event sponsored by the University of Texas at Austin. The views expressed tonight are of the student organization, invited guest speaker, and do not represent the views of the university or its officers.
Thank you, Emily. My name is Catherine, and I'm a sophomore biology track environmental science major in Dean Scholars, and I'm so grateful to have co-chaired with Ashley Hoffman last fall with Dr. Susan Solomon and Emily May this spring with Dr. James Allison. This year, we are extremely honored and excited to host Dr. James Allison, a UT alumnus and the 2018 Nobel laureate in physiology and medicine. Most noted for discovering CTLA-4 and checkpoint immunotherapy, Dr. James Allison has both pioneered a new field of research and revolutionized our understanding of cancer therapies. Born in Alice, Texas, James Allison received his bachelor's degree in microbiology, and then in 1973, his PhD from the University of Texas at Austin. Allison started his career at MD Anderson in 1977, arriving as one of the first employees of a new basic science research center located in Smithsville, Texas. Afterwards, he joined the faculty of UC Berkeley. In 2006, he moved from Berkeley to Weill Cornell Medical College. He was recruited back to MD Anderson in November 12th to lead the immunology department and establish an immunotherapy research platform. His research has centered on elucidating the mechanisms by which T cells recognize foreign particles, identifying molecules required for T cell activation. And in 2011, under the trade name Yervoy, the antibody developed for cancer treatment became the first immune checkpoint therapy to be approved by the US FDA for the treatment of late-stage melanoma. In addition to the Nobel Prize, Allison has been the recipient of numerous other awards, including the 2014 Canada Gardner International Award and the 2014 Louisa Gross Horwitz Prize. He was an elected member of the multiple organizations, including National Academy of Sciences, the American Association for the Advancement of Science, and the National Academy of Medicine. Without further ado, we are honored to present Dr. James Allison's lecture, Immune Checkpoint Blockade in Cancer Therapy, Historical Perspective, New Opportunities, and Prospects for Cure. Dr. Allison? You're muted, Dr. Allison.
Glad to have this opportunity to give this talk. As was said, I am an alumnus of UT Austin. When I was there, I'll skip this. I don't need this, I don't think. It was when T cells were discovered, actually, it's a long time ago. And having a little trouble.
Dr. Allison,
Yeah.
You may be able to click on the slide to advance. There we go.
Yeah, there we go. Anyway, when I was in UT Austin, I was an undergraduate. I was actually studying biochemistry with Barry Kiddo, who recently retired. And I had the good luck to take a course in immunology from Bill Mandy, who unfortunately passed away many years ago. But he taught us a course in immunology. And right at the end of it, T cells had just been discovered. And he said, there are these cells that go around the body and go through the tissues, go through the blood, go everywhere in the body and look for stuff that, you know, shouldn't be there and handle them. They do the right thing. They either alert other cells or they kill them or whatever. And I just was fascinated. I said, how do they work? And he said, we don't have a clue. Nobody knows how they work. Nobody knows how they recognize them. Nobody knows anything about that. And so I decided at that time that I was going to study them. I was going to work on T cells, you know, for the rest of my life. And so hence the talk I'm going to give you now, which talks about how we figured out how to mobilize these cells to attack cancer. Most of the work that I'm going to be talking about tonight was done by a very talented postdoc named Spencer Way, with contributions from the rest of the people that are listed on this slide. But I just want to show you, just to start off, that this actually works. This is a photograph of a CAT scan of the brain sorry, this is the lungs of a patient who had metastatic melanoma, stage 4 melanoma. At the time, this was 2004, the life expectancy after diagnosis was 7 months. Anyway, you could see some of these tumors circled in red in her lungs. And after the therapy in 2006, they completely went away. And this one shows she also had a metastasis in her brain. It was 1.5 centimeters. And that also went away. She was completely cured. And she now is, you know, this is old, obviously, but she has 2 beautiful teenage kids that she's managed to bring up. So I said she was 22 years old, just about to begin her life, and was told that she was, you know, had 7 months to live, and she's still alive today. Anyway, she's become a very good friend. And anyway, so this came out of work that I did starting, as you heard, at Smithville, when I was just decided to study how T cells recognize antigens and how they're regulated. And one of the things that we discovered was that, you know, there are 2 signals needed to activate T cells, not just the antigen, not just recognition of antigen, but also a co-stimulatory receptor. And so tumors can have all kinds of foreign things on their surface that are there because of mutations. And the immune system doesn't see them because they're invisible, because they can't give that second signal. So the only way that we showed this in the early 1990s, that T cells recognize them, is by tumor cell death. And a particular kind of cells called dendritic cells come in and phagocytize the tumor cells and then redisplay on their surface the antigens. And it gives a T cell receptor signal, along with the signal that we showed in 1988 was mediated by CD28. It's a necessary second signal to get T cells activated. But then we discovered after about 5 years that there was another molecule called CTLA4 that was a very, very close cousin of CD28 that gets turned on when you start the turning on process, because you've got about 100 million different T cells. So you've got a few dozen against any particular antigen. And so when they detect antigen, they've got to start proliferating and expanding the clones to give you the army of tumor-specific, antigen-specific T cells to get out there and actually, you know, do the job. And so they have to proliferate, but that has to be stopped. And so CTLA4 stops that process at a point, or it will kill you. We made knockout mice that lack CTLA-4, and they died when they're about 3 weeks old because their immune system just starts proliferating to all the things they're exposed to, and it can't stop, and they just fill up with T cells. And so I had the idea that maybe we could just take the brakes off because it's a race because the tumors have a head start and they're not recognized until they reach such a mass that they cause inflammation, alert the immune system, that it's a race between when CTLA-4 comes on and stops the T cells. And if they do that too early, the tumor wins. And so I had the idea, let's just take CTLA-4 out of the way. Let's just let T cells keep going for as long as we need them to eliminate tumors. And that's what Sharon Belvin, she showed in the last slide, happened. And there's this compelling notion for a couple of ideas. One is that you're treating the immune system. You're not treating the tumor cell, you're treating the immune system. And so potentially this can work for any kind of cancer. It doesn't really matter. There are limitations that I'll talk about in a moment. But in any event, potentially it could work against them. And also because it starts with necrotic cell death normally. If you just give monotherapy with this anti-CTLA-4 antibody we had, but also it has the potential to give T cells, to give other therapies the benefits of immunotherapy. T cells, once you've got them for the rest of your life. And they can come back and they can recognize tumor cells. And if the tumor comes back, they can eliminate it. Other therapies like chemotherapy, radiation, hormone therapy, whatever, if you don't kill the last tumor cell, the tumor wins. But with immunotherapy, you can initiate your own body to start attacking the tumor cells and do a much better job. Actually, was the idea. And so this was one of the mouse experiments that we did. We did many of these. And this is just showing the growth of a tumor. The black line is just an untreated tumor growing, injected on the back of mice. And it grows, and you have to euthanize the mice after a while because it gets too big. If you block CD28, which is that co-stimulatory signal, the tumor actually grows faster, showing that there is a nascent immune response to the tumor, but it just can't get the job done. But when we injected anti-CTLA-4 antibodies that we'd made in the lab, the tumor grew for a little while, and then after the immune system got clued in that there was a tumor there and you got enough cells generated, the tumor was rejected. And those mice, for the rest of their lives, were immune to re-challenge with the same tumor. And so this, gave us an idea that this ought to go into humans. There was a lot of skepticism, but just to cut to the chase, we finally teamed up with a company called Medarex that had developed some mice that had the immunoglobulin genes, the antibody genes, replaced by human genes. And so you had a fully human antibody because if you injected a mouse antibody into people, it would lead to problems because the people would make antibodies to the foreign protein. So anyway, we made this fully human antibody to CTLA-4 and it went into trials. And there were objective responses, as we had predicted, against many tumor types, including melanoma, prostate, kidney, bladder, a lot of other cancer types. There were, we found when we went into patients, into humans, there were adverse events which we never saw in mice. And we're not completely sure the reasons for that. But anyway, there are a lot of itises, inflammatory diseases, from really bad diarrhea to inflammation of the liver, to problems with the adrenal glands. But they're generally manageable now. Doctors have worked out algorithms for dealing with them. And although there are very rare autoimmune things that happen, there's no free lunch when you liberate the immune system to attack cancer cells. So it's been shown now that a fraction of, it's less than 1% of patients develop diabetes or myocarditis. But we're working on it. We've got a model now. I don't have time to talk about it. And we're working out ways to do this. Anyway, in 2011, almost exactly 10 years ago, this week, there was this paper published, and FDA approved this drug. This shows the survival of patients that got a placebo. That, again, mostly they were with stage 3 and 4 melanoma. They're mostly dead within 7 months or so. And this line here is the CTLA-4 line. The patients that got CTLA-4, we got 4 doses at 3-week intervals. So they got treated for 3 months. And you can see that survival curve flattens out. And in 2015, there was a follow-up. By then, there were over 5,000 patients of whom there was a 10-year history. And that line flattens out at about 22%. So about 20 to 22% of patients with metastatic melanoma are cured. They're alive for 10 years plus. There is a woman from the phase 1 trial in 2001 that is still alive. They got a single treatment with this drug. So it's a very powerful, very curative drug. And so, but the question was, as a scientist, I said, why just 22%? Why isn't it everybody? Because the wiring of the immune system is pretty much the same. Well, there are a lot of reasons for that. I could give you some mechanistic reasons why, if you don't have the priming going on while CTLA-4 is on board, it's not going to work very well. But the other possibility, which is a lot more interesting, of course, to a scientist, is that maybe there's another checkpoint. And so Tasuku Hanjo in Japan discovered another checkpoint in 2001, as our clinical data were coming out. And this one works slightly differently. PD-1 is a molecule that's expressed on T cells, and it binds to a molecule called PD-L1, ligand 1, which is induced on tumor cells when the T cells are attacking it. They make gamma interferon that upregulates expression of this gene, and so it's an adaptive defense mechanism. So it's very different than the way that CTLA-4 works. But anyway, it went into phase 1 trials. It was also, again, because you're treating the immune system, was effective against melanoma with 30%, close to 30% objective responses in melanoma, and 18% in non-small cell lung cancer, 27% in renal cancer, colorectal cancer. There weren't any responses, but in the phase 1, but later it was shown that there is a subset that has a lot more mutations because they have defects in DNA damage repair. And so they put out a lot more mutated peptides. And also castrate-resistant prostate cancer didn't respond whereas it did to CTLA-4. And I don't think I have time to talk about it, but we know the reasons for that now. But that these were independent pathways was shown was that patients that failed treatment with ipilimumab could go on to successful treatment with nivolumab, which is the PD-1 antibody that Bristol Myers Squibb made. And vice versa is the same. Patients that failed PD-1 could respond to ipilimumab. And so this, confirmed what we had worked out from mechanistic views, that these were different mechanisms. And so the obvious thing to do then was to try combination therapies. And so that's what was done. So we did this first in mice, showing that these were additive. You can block PD-1, PD-L1, and CTLA-4, and if you blocked them all, you got the maximum response. So these were at least additive. So this led to a trial in humans in melanoma again. And this just shows, this actually is not the most recent data, which has not been published, but this just shows the 1.5 year survival, which is at 55%. But now we know from following these same patients that this is still true. Over 50% of patients that get this combination therapy are alive 5 years plus. And so there's not any reason why they're not going to, go out to, 10 years, like with ipilimumab only. And so this is a curative therapy for this, which was a devastating disease, and the first one that we tried it in. So what worked out in subsequent years is that the number of mutations in different tumor types varies. These here on the right hand of this slide, lung cancer, bladder cancer, melanoma, these are caused by external mutations. Melanoma by ultraviolet radiation. These others by carcinogens that are found, in smoke. And all of these respond very well to monotherapy with CTLA-4. This shows the mutational levels. But it works even down lower with kidney cancer, which we don't exactly understand, but also in prostate cancer. As yet, we have not had any success in pancreatic or glioblastoma, but this is something we're working on. And MD Anderson at the immunotherapy platform. But it's not just melanoma in these drugs. This just shows the different kinds of cancers that these drugs have been approved for the treatment of. Melanoma, lung cancer, kidney cancer, colorectal cancer, it just goes on and on. Something over 50 approvals now by the FDA, which means that any doctor can prescribe these drugs, CTLA-4 or PD-1, the combination, to treat these kinds of cancer. And it's expanding every day, and we're making improvements to it. So, as we had hoped, it's not just limited to melanoma. But what I want to do now is really get deeply into the science, because that was one thing that I learned when I was at UT Austin working, is that, there's nothing like, really being reductionist and studying mechanisms. These two different drugs work very differently. So I told you CTLA-4 is hardwired. It's involved in every immune response that you get, no matter what the challenge. PD-1 is induced resistance. It's in the tumor makes PD-1 when it's activated, but it combines with PD-L1, which is induced by something that the T cells make. And this mechanism is involved, it's been shown, in protecting the embryo against destruction by the maternal immune response to, paternally induced antigens. It's one of the defense mechanisms that protects the embryo, and tumors have somehow adopted that to do it. CTLA-4 targets the CD28 co-stimulation pathway, and PD-1 seems to target T cell receptor. CTLA-4 works during priming. PD-1 works on totally differentiated T cells, as I'll show you in a moment. So very different effects. CTLA-4, because it works during priming, can recruit new clones to an immune response, whereas PD-1 only works on expanding clones that were already generated, and does not really recruit any new ones. Anyway, some other stuff, as I'll show you, CTLA-4 primarily affects CD4 cells, although CD8s as well, whereas PD-1 only affects CD8. These are the killer cells for those of you who haven't had immunology. And we showed a few years ago, Pam Sharma in the immunotherapy platform showed that CTLA-4 blockade can actually move T cells into cold tumors such as prostate cancer, which don't really have any T cells in them at baseline. Whereas PD-1 can't do that. It only expands cells that are already in there. CTLA-4, perhaps because how it works, has a lot of adverse events, more than PD-1. So PD-1 is used a lot more regularly than CTLA-4. But the trade-off is that recurrences with treatment with N-acetyl-A4 after responses are very rare because of these other issues that I've talked about earlier. Whereas about 15 to 25% or so, depending on the kind of cancer, do recur after responses to PD-1 therapy. And so we wanted to figure out, what are the differences between these? What are the cellular impact of these two different therapies? So that we took advantage of a high-dimensional approach called CyTOF, where you use antibodies that are coupled to rare earth isomers. And so you stain cells with the antibodies coupled to these heavy metals, and then they go through this nozzle and they get subjected to mass spectrometry. And so we can measure up to 43 things at a time and really get an idea of what's going on. So this is kind of what the total map is when you use algorithms that group cells according to how similar they are. You can see myeloid cells here. You can see PD-L1 plus, hematopoietic cells here, regulatory T cells here, CD4, CD8, all of these. And so we did a series of experiments where we treated mice and then harvested the tumors at a point where they were just beginning to regress and subjected to this kind of analysis to see what was going on. And we were most interested in T cells. So this just shows the schematic of the experiments. The mice were given tumors, and then at day 13, when they were after treatment, when they were beginning to regress, and we've done this with a number of different cancer types, and it doesn't really matter. The mechanism seems to be the same in all treated with PD-1 or CTLA-4. But this is what it looks like. Again, this is if you look at untreated mice and just look at all the cells. But if we gate for T cells using a marker, CD45, meaning hematopoietic cells, and then CD3, which picks up a molecule that's just on T cells, this is what it looks like. And you can see here that in blue are the untreated mice. Red is CTLA-4. These are dot plots. So each dot represents a cell. And PD-1 is in green. You can see here that Tregs are depleted by CTLA-4, not by PD-1. But the number of CD4 effector cells has increased by CTLA-4 treatment. And if you look at CD8, these are the killer cells of the immune system that can destroy foreign cells. And you can see here that there are patches of different ones that are affected by CTLA-4 or PD-1 differentially. And then there's other kinds of cells, immune cells called NK T cells and gamma delta T cells that they don't really change at all. So they don't really play a role in the anti-tumor response. So this is called a phenogram, and this is a heat map of individual molecules according to the different clusters that we detected. And this is just not meant to confuse you. It's just to show how confusing this data can be. When we broke it out, this is what we saw. Again, as I told you, CTLA-4 actually depletes these regulatory T cells, which are the ones that inhibit T cells, the ones that actually eliminate tumor cells. This only happens in mice. We've not seen this in humans, which is a little confusing, I know, for this lecture, but it's just a special property of the antibody we made. But if you look at CD4 T cells, these helper T cells, there is this one population that we call Th1-like. These express a molecule called ICOS, that I'll come back to in a moment. And also because they're activated, they express low levels of this molecule called PD-1 and also a transcription factor called TBET, which enables them to make gamma interferon and TNF-alpha, which can kill tumor cells. That does not happen with PD-1 treatment. And there are naive T cells in there that are not increased by either treatment. But if you look at the CD8s, you see these two populations here that are differentiated T cells. These in particular are interesting because they are the ones that actually have really killer, they can kill cells. They're increased by CTLA-4 and also shown in green by PD-1. But if you look at PD-1, it increases the frequency of these cells that express PD-1, TIM3, TBET, and LAG3. These are cells that are exhausted. They're named that because they've been killing tumor cells and they don't function very much anymore unless you give the antibody to PD-1 and then they expand. And that's why this numbers went up. But if you quit giving PD-1, they will shut down again. And so PD-1, another difference is that it's given for as long as 2 years to patients, where CTLA-4 is only given, initially. Anyway, and the rest of this stuff I'm not going to get into because it's not interesting. But so anyway, the bottom line is that CTLA-4 blockade gives you an increase in these interesting CD4 T cells, and these two types of CD8 T cells. PD-1 increases those two, and this will become important later. And so anyway, this just summarizes that the monotherapies differ. They share this because these terminally differentiated killer cells are expanded by both, but CTLA-4 expands these CD4s, and in this situation at least does not expand these CD8 exhausted cells that have these 3 different inhibitory molecules that were discovered after we discovered CTLA-4. CTLA-4 was the first inhibitory molecule that was discovered that we found in the mid-90s that inhibits T cell activity. But ICOS is very interesting because it's a member of this gene family, CD28 and CTLA-4. Phylogenetic tree is here. It's closely related to CD28 and CTLA-4. It has its own ligand, ICOS ligand. But at the time this was found on T cells in tumors, it was really interesting because immunologists would say it's primarily associated with follicular T helper cells, which are only in lymph nodes, in association with B cells and helping them do class switching, or a special form of regulatory T cells, CD4 T cells. But Pam Sharma in 2006 did a pre-surgical trial in bladder cancer of patients treated with CTLA-4 and found that there was a huge increase in the number of these ICOS positive, TBET positive gamma interferon producing CD4 cells, uniquely expanded by CTLA-4 and not by PD-1. So it's a very interesting class of T cells. And so these are just some of the studies that she did in her early clinical trials, showing that these cells were a marker of anti-CTLA-4 activity. And then later, mouse studies showing that if you try CTLA-4 therapy in mice which have eliminated ICOS or its ligand, the therapy does not work nearly as well. And she went on to show that what you need is a PI3K binding motif, and that signaling through that motif makes T cells make more gamma interferon. And then we also got together and patented an approach where we targeted ICOS and could increase the efficiency of CTLA-4 blockade. So this, as a basic scientist, got me interested in thinking about, if you block negative co-stimulation, does this regulate how T cells develop? And so what we did was we went back and did this whole high parameter mass spectrometry analysis on wild type mice, but also on CTLA-4 knockouts and littermates. And there's 2 different ways of expressing the data, which I will show you. One is just by grouping cells according to what's more similar, and the other one is tracking cells according to how mostly different they are. So anyway, this is what it looked like. So when we knock out CTLA-4, these red populations are cells that they're not there in wild type mice. We also could not find them in patients in their normal blood cells until they had gotten CTLA-4 antibody. That was the only way you got them. Or in mice, you had to treat them with the antibody or knock the gene out. And then you got these new kinds of T cells here, like that one. It's like Australia. It's very different. The cells down there are very different than these other cells and have a lot of similarities. And so if you look at these, there are these different kinds of cells that, again, if you look at the blue dots here, that's the frequency in wild type or heterozygous, as opposed to the red, which are the ones that appear in the knockouts. And so these 4 peaks are new cell types, plus a little bit of this kind, that appear only when you get rid of CTLA-4. And so these guys here are exactly those cells that we see when we're treating with the anti-CTLA-4 antibody. And these others are intermediates that occur along the way to full differentiation of T cells. And I'll get to that in a moment. But the thing about T cells is they develop, a CD4 T cell comes out of the thymus, and it's got a receptor, and it's got the CD4 co-receptor, but it has no function pre-programmed until it gets activated by antigen. And so depending on the strength and a lot of other things that are going on in the environment, it gets programmed to become a different kind of T cell. And so we think that what's happening is that CTLA-4 controls this and limits the expansion, the phenotypes of T cells that can be developed when they're developing in the absence of CTLA-4. It just compresses them. And so this is the classical view of T cell development. If a cell ends up getting BCL6, it's a follicular helper cell, GATA3, depending on the signaling pathway, it makes type 2 cytokines. TBET, the one that I talked about earlier, is associated with gamma interferon production and TNF alpha production. Th17s come from getting ROR gamma T. They make a different kind of cytokine. And finally, FOXP3 positive cells with that transcription factor suppress the rest of the cells. And as I shown you earlier, one of the cells that we saw when we block CTLA-4 or knock it out genetically has 3 of these transcription factors, and they're making all kinds of cytokines. So they're really confused. And so there was work from the late 70s when these different phenotypes of CD4 cells were developed that showed that signal strength can play a role in dividing them in their development. And so the ones who have the strongest signal strength tend to be Th1s. And so what we think these data indicate is that CTLA-4 normally puts a constraint on the signal strength, because you need T cell receptor, you need CD28, and CTLA-4 attenuates CD28. And so it lowers the overall signal strength. And so it keeps T cells from going to those extremes. But when you take it away, either with the antibody or genetically, you give rise to these new T cells, which I just showed you. And so we think that this is what goes on, because CTLA-4 works during priming, and it constrains the possible differentiation pathways that CD4 T cells can go down. And so this suggests that it's not just this simple linear progression that people have talked about. It's more complicated and has to do with multiple signals and with strength of multiple signals to get to these. So it's more of a variegated developmental pathway. But by giving this drug, anti-CTLA-4, we really favor getting Th1 cells. So it's not that you're just expanding T cells that were already there. You're creating a new kind of T cell that actually wasn't there before. And if you do the same thing with PD-1, you don't see any of that. PD-1 does not, because it works on terminally differentiated cells, it does not give you anything new. And so one of the questions that we had was, when you put these two mechanisms together, are they just additive? You know, is it you're doing the things that CTLA-4 does plus PD-1, or is it maybe something different? And so we went back to that same kind of analysis that I showed you before. And so what you see here is that, again, with control mice or CTLA-4, these cells that have these three inhibitory signals on their surface, PD-1, LAG3, and TIM3, they expand only when you give PD-1. But the other cells that are less refractory, that have very low levels of LAG3 and TIM3, they don't expand so much when you give the combination therapy. In fact, they go away. These terminally differentiated, exhausted cells actually go away. And so not only do they go away, but if you do a plot of the number of cells versus tumor shrinkage, which we did, they become irrelevant. They're not associated with tumor treatment anymore. And so what happens? Well, they're replaced by these cells, which are terminally differentiated, but not terminally exhausted. These are different because they're activated. So they have PD-1, they have TIM3, but they don't have LAG3 at high levels. And when you give CTLA-4, in addition to PD-1, these cells go way up and they replace these different. So the mechanism changes fundamentally when you give the two drugs together. And these are the cells that matter. And then also, because activated CD4 cells have PD-1 on them as well, when you give the combination therapy, they expand more with CTLA-4, which creates them, and then PD-1, which can limit their expansion. So we get an increase in those cells as well. And that's why the combination therapy works so much better. So this just summarizes it. You go from these differences to simplifying when you put them together. You just get these ICOS positive CD4s and these terminally differentiated but not exhausted CD8 effector cells that can kill tumor cells. And that's why the combinations work better. So what happens to those exhausted cells, we don't really know. One is that they may be converted back into effector cells, but a number of laboratories have shown that there are epigenetic changes in those cells that are thought to be irreversible, so they can't go back. And so we think that maybe the exhaustion of the effectors are prevented when you allow them to get more abundant CD28 co-stimulation by taking the inhibitory effects of CTLA-4 out. So these are things that we're actively testing in the laboratory. And so just to close, since we discovered CTLA-4 and Hanjo discovered PD-1, there are a lot more molecules that influence the various things that immune cells do. And all of these can potentially be targeted to do something about the cancer. And so the what's going on now in the clinic is blocking multiple checkpoints. I've shown you can block negative ones or positive ones with the ICOS. Also, you can add these to radiation and chemotherapy to give them where you don't have to kill that last tumor cell. Because as mentioned earlier, the only way that chemotherapy or radiation works is if you kill every tumor cell. And in order to do that, you have to do a lot of damage to normal cells, and also genetically targeted therapies that inhibit enzymes that drive cancer. There's also a small number of trials that have been done to show that those work in combination. So this is the effect of this. I mean, for the last 30 years, since the advent of chemotherapy, really, this has been the goal of oncology, of treating cancer, is that you treat large numbers of patients with the drug, and then you see if there's an increase in the median survival. And if there is, then you call it a success, even though it may be a few weeks or a few months or a few years maybe. CTLA-4 did that. It increased the median survival by 4 months, but also, as I showed you earlier, that survival curve plateaued out at about a little over 20% of patients who had no recurrence after a single round of therapy. And so this next line is not data, but it's our aspiration now. This is our aspirational target, to move that tail on the survival curve as high as we can get it, and as many types of cancer as we can get it, to cure more people with cancer of many different types. And so with that, I will stop, and we want to take questions. So I will stop screen sharing. I don't know exactly what to do now, but.
Hello again. Thank you, Dr. Allison. That was a really wonderful talk. And so we have a couple questions. One of the questions that we got was, what other mechanisms of cancer-related T-cell suppression do you believe show promise right now, or that could be used as additional treatment strategies to unleash even stronger T-cell responses?
Well, one would be getting rid of regulatory T-cells for sure. And the other is that, I'm a T-cell person. As I said, I got interested in T-cells at UT Austin. But the tumor microenvironment has a lot of cells, a lot of different kinds of cells, including myeloid cells, which are very different. They're macrophages and different types of cells like that that can also be suppressive, but they can be changed actually to help an immune response. And so that's one of the things we're working on, is to figure out how to get rid of the suppressive myeloid cells and increase the number of the anti-tumor myeloid cells. So there's a lot more to be done, a lot more science that we need here.
All right, awesome. And what is the role of genetic testing in determining who will respond favorably to immunotherapy?
We've not really found anything yet. There's nothing in the genotype that anybody's found that predicts who's going to respond. So that's still a bit of a mystery. No single marker, I should say, although there are combinations of markers. If you look at the tumor and the individual that has it, there are some that are beginning to play out, but it's nothing simple like we would have hoped. No way of before treatment predicting who's going to respond by any kind of genetic test. There's been a lot of effort to look at that, but it just hasn't proven out. We can know after one dose of antibody who's going to respond or likely to respond, but at baseline, unfortunately, there is no test right now. So it's a black box.
Next question, is the expansion of the CD8 effectors in the presence of combination therapy due to the new quote unquote helper T phenotypes created by anti-CTLA-4 therapy?
Yeah, in part, that's a very astute question. But yes, in part, I think that those helper T cells they not only make cytokines which can kill tumors themselves, but they also make cytokines which can help the CD8 cells become more cytotoxic. And so I think it's a combination of the two.
All right. And so what are your thoughts on immunotherapy being an effective treatment for neurodivergent diseases like Alzheimer's or Huntington's?
Yeah, that's a difficult question. I think that there is evidence that those are partially, autoimmune treatments. If we could find some way to turn specific T cells off, that might be useful, but it's way too early now to say anything. We don't know how to specifically turn, antigen-specific T cells off. We can unleash them, but we can't turn them off yet.
As a follow-up, how would this work if they aren't necessarily genetic, or do you have any thoughts on that?
What do you mean, genetic?
Sorry, I misread the question. How would this work if they are genetic?
Oh, I think if the question is in certain kinds of cancer, like BRCA1 mutations that predispose certain, women in particular, and men as well, to cancers, there are possible ways of that are being thought of. I mean, they're not perfected yet, but of prophylactically treating that to try to keep people from getting cancer. Other than that, there's not a lot of insight.
Next question: could inhibition of CD28 be used to treat autoimmune diseases?
Yeah, it can be, and you have to be careful with it though, because if you do the wrong thing and give a signal through CD28, it ignites all of your T cells. So, but blockade of CD28, and the way that it's being done now in the clinic, it's the most successful, is actually making a recombinant form of the CTLA4 molecule. Because what I didn't talk about is that CTLA4, one of the reasons it works to shut down immune responses, it binds to the B7, it binds to the same ligands as CD28, and it binds much tighter. So Bristol Myers Squibb and some other companies have made recombinant forms of the CTLA4 molecule itself, which could stop priming and is useful in treating rheumatoid arthritis and other autoimmune diseases.
Switching gears more towards just STEM in general, do you have any advice for current undergraduates? And what is something that they should be doing right now to help increase their chances for success in future STEM careers?
That's a question I get all the time. And all I can say is follow what you love. If you're really interested in something, if you're really passionate about figuring out how things work, if you really like science and math and all that incurs, do it. Just do it. It'll be the best that you can at it. Just follow your passions. I mean, that's all I could say is, and go to a, good lab which allows you to do that, and it'll all work out. At least that was my history.
Following up with passion, what motivates you to keep pursuing your questions? Scientific research can be discouraging sometimes. What helps you keep going when you don't have necessarily just concrete results in front of you?
Yeah, that also is something that's worth talking about. And once you've been in science for a while, you realize that you can go for years, literally. I mean, it was 1994 when we first had the idea that CTLA4, might be useful to treat cancer. It was 15 years later before it actually made it in the clinic. But anyway, thing is that there's no instant gratifications. You just have to keep plugging away and have this satisfaction that when you make a discovery and you find something new, that's worth all the toil and the pain. You just got to get used to that. There's a lot of what you're going to do is, science is, people have this feeling that, science is magic, that, the scientific method is you make a hypothesis and you test something and you get a right answer or a wrong answer, but that's not really how it works. Most of the time you're wrong. So you have to come back again and again and again and just be persistent, and be curious. So that's why I say it's passion that has to drive you, otherwise, I think that sometimes I envy my colleagues that, are physicians, because, now at least, they can treat patients and have instant gratification because they see responses and stuff. Science doesn't work that way. It goes on for years, and it's very frustrating, but that's just part of it. If the answers are easy, they're probably boring and somebody's already found them out, so you dive deep in it, it becomes harder, but that's what you should do. Dive as deep as you can and pursue your passion and figure out what you want to do, what you want to know.
I suppose following up to the question about, or the response about diving deep, what was your sort of diving deep moment where you found a turning point in a defining moment in your scientific career, like really diving deep and your turning point in that scenario?
There were several actually, but maybe the best one was when, we figured out, which, nobody had shown before, that the CTLA-4 molecule inhibited T cells. And at the time, people thought, oh, T cells get activated and they divide, like as I was saying, they go from a few dozen to hundreds of thousands, over a week. And then people said, oh, they just die. And, that's not how it works. That's pretty boring when you think about it. But CTLA-4 is what controls that. It tells the T cells to stop proliferating. And so we got that. And then I thought, now we can treat cancer. So we first did the experiment after we made an antibody to CTLA-4, and then treated mice with cancer. And I thought, and I saw the tumors go away. I was like, wow, okay, we're on to something big here. The intellectual reward was figuring out, that the T cell receptor was kind of like the ignition switch, and CD28 was the gas pedal, which we did in the late 80s. And then CTLA-4 in the mid 90s, we said, that's the brakes. So we disable the brakes and we can treat cancer. And so that was the big one, I guess, for me.
All right. As a follow-up question to close the night, our final question, what is the most rewarding part of your research?
I think that, again, I since I've been a little kid, I've always wanted to figure stuff out. I was one of the guys who would take apart a radio and never could put it back together, but I just wanted to figure out how it worked. And then I got into science, like I said, because of this immunology course I took at UT Austin. Like I said, there's these T cells and nobody understands anything about how they work. But the most rewarding thing is meeting patients that are alive because of these therapies that came out of that basic science. So to me, I'm just so lucky, because I didn't get into this to develop any treatment of cancer. I got into it because I wanted to know how T cells worked, and the fact that a few 100,000 people have been treated with these drugs now, and 30 to 40% of them are cured is the reward. So I don't think everybody could be so lucky, but I was.
That is a very nice reward, and that's a very heartwarming place to end our discussion with. And thank you so much, Dr. Allison, for all the time. And thank you to everyone on the live stream who watched us tonight. And thank you to everyone involved with making this possible. And we hope to see you for spring 22 DLS. Thank you.
Hope I get to Austin.
Yeah, hopefully.
Okay.
See everyone.
Okay, bye-bye.